There we go. So I'm going to take you through, I'm going to give you some genetics 101, so strap on and get ready for this. We're going to go through a whirlwind of some new things that we're doing in genetics, genomics, and technology development that actually does impact how we can and do think about genetics in Duchenne. So Duchenne muscular dystrophy is one of the most common rare genetic diseases of childhood. This is the progression of a boy, and in all openness, this is my son who actually has Duchenne, and he's 18 years old now. Duchenne is the 800-pound gorilla in the neuromuscular space. So it dominates sort of shortened lifespan for muscular dystrophies, and it's sufficiently common that we can study it well. So it has a lot of sort of special place within sort of the broader neuromuscular world, or even the broader world of rare genetic diseases. There's over 5,000 such rare genetic diseases that we've already described as a community. There are thousands more yet to discover. Some of them will be ultra rare. Some of them will occur in a single child, and we've already seen that along the way. Duchenne is not that sort. It actually affects a couple 100,000 people in the world, and there's thousands born every year with Duchenne muscular dystrophy. The gene was identified that's causal in 1986. It's on the X chromosome. The gene is huge. Because the gene is so huge, it's a mutational target, just a large portion of the genome. So 1 in 10,000 cell divisions mutate the DMD gene. That's sky high. It's much higher than a typical gene. And that's the basis by which it's so common, coupled with the fact that it's on the X chromosome, so boys only get one X chromosome. So that combination leads to the very high rate that we see in humans. So it's an X-linked recessive disease, so it mostly affects males. If you have a loss of function mutation, that is, if you don't make any dystrophin protein because of a DNA mutation and you're a male, you will have Duchenne muscular dystrophy or a variation of Duchenne muscular dystrophy. Typically, women who are carrying one mutant allele and one non-mutant allele typically don't have any signs or symptoms, although we heard about the cardiac symptoms a bit ago, and we do have carrier women who are overtly affected with Duchenne. We actually have girls with Duchenne muscular dystrophy and everything that's in a spectrum between completely unaffected to overt Duchenne muscular dystrophy. So take the X-linked recessive with a slight grain of salt, but that's its basic genetic mechanism. And what it means is that, where am I going to do? There we go. That it typically is observed just in males, and that this brother of this woman got the disease and also has one of the mutant X chromosomes. This woman has an X chromosome that has a mutation and a normal, and the X chromosome that was abnormal with the mutation was passed on to this man and not to this man, and therefore this man's affected. So it's transmitted through females, and males are affected. So that gives us lots of information in terms of how we go about doing counseling in clinic, and I'm sure many of you have received very good counseling. One of the things that we know very, very well now is the disease is completely driven by new mutations. It occurs all over the world. It occurs in all populations. Every human population is at risk for Duchenne muscular dystrophy, and the spectrum of mutations look exactly the same. So we can go anywhere on the planet. Any population, as far as we know, will always have a very similar distribution, and we know this from a number of studies that are actually keep coming out from many different countries across the world. Almost all families actually have their own individual mutation, which is a little bit of a head scratcher, right? So, somebody else has an exon 45 deletion. They've got my same mutation. The reality is they don't. They actually have an independently occurring exon 45 deletion, and that's a distinct mutation from another person with an exon 45 deletion. We call it the same in terms of how we report it out clinically, but they're actually distinct mutations with distinct boundaries of the mutation. And that's, again, driven by this high mutation rate. So the breakpoints for a lot of these mutations are actually distinct for the most common. The other thing which is really rather unique about Duchenne muscular dystrophy is because of the high mutation rate, it's driven by de novo mutations. And what that means is it's almost always a surprise in every family It doesn't typically exist in the family. The first thing you know about it is your son is affected, and the mother may or may not be a carrier. If the mother is not a carrier, then it means that there's a de novo mutation in that child, typically a boy, and the mom is by definition, some slight exceptions to this, but by definition a germline mosaic, meaning a portion of her eggs are affected, and one of those eggs made this child. But she does not have a mutation in her blood, and she has no risks. She also, if she doesn't have the mutation in her blood, there's no risks to her sisters or her mother in terms of being a carrier, but there are risks to her female progeny. So this is in our clinics, typically a third are inherited, and we knew that the disease existed in the family. A third are de novo mutations that occur just in the child, and the mom is not a carrier. And a third, the mom is discovered to be a carrier only because we diagnosed her son, and then we work our way back up the family to figure out where did the mutations start. It always starts somewhere, and it always starts in a really short time history in every single family that we look at. So there's been some shifts in the diagnostic process throughout, looking around the room, most of our lifetimes. In the past, before we had DNA diagnostics, it was really history, physical, a clinical diagnosis, get a CK level, it would be sky high. You'd then get a muscle biopsy, and the muscle biopsy would show a dystrophic process, and you'd be diagnosed with Duchenne Muscular Dystrophy. After the gene was identified, the next step was that you could do DNA analysis from blood and prove that you have a mutation in the DMD gene and not need to necessarily do much additional work. This also allowed us to make protein markers for Duchenne muscular dystrophy that can be used on muscle biopsies that allowed us to prove that the muscle does not express dystrophin. So when we do this testing now, we now have a DNA mutation that can be used and tested on other family members to figure out where the mutation occurred in the family and assess genetic risks to subsequent pregnancies in other family members, as well as the family that came in for genetic testing. All patients, all of you, anybody you know, anybody you might know in the future, needs molecular genetic testing. It's actually critically important at this point. Many clinical trials and actually some drugs which do have a benefit depend on knowing the exact mutation, but every family needs to know this in terms of knowing their risks. Some examples are nonsense mutation. Patients are amenable to a nonsense read-through drug. Some deletions are amenable to eteplirsen, which is FDA approved. There's other exon skipping trials in progress, which may lead to new FDA approved drugs, which again, we'll need to know the exact deletion. Some mutations may exclude you from some of the gene therapy trials. Some may include you in gene therapy trials. So, all right, this is your lesson. Get ready. This is fun. This is actually genetics is amazing because it's universal. The simplest organism that exists on our planet uses exactly the same genetic code with very minor modifications as we do as humans. So a lot of what I'm going to tell you about is the same in bacteria, yeast, fungus, viruses. So when in the 60s, once DNA was figured out to be the basis by which material is transmitted from father to son, mother to son, et cetera, it was recognized that DNA made RNA, and that RNA used a 3-letter code to determine which of about 20 different amino acids are encoded by each RNA letter. So a TTT encodes an amino acid called phenylalanine, an AAT encodes a different amino acid, a GGT encodes a glycine. So you have this code that everybody uses. It's a universal code. We can actually take a bacterial protein and express it in a human cell. We can take a human protein and express it in a bacterial cell. It's the basis of a lot of drugs right now. So the structure of the genome is complex. So genes are typically expressed as exons. So a lot of you have been told your mutation is a deletion of exon. Give some numbers. And this is what that means is exons are the protein coding part of the sequence. They have little edges here that allow them to join to a next exon and the little sequence here that allows them to join here. All of these introns are a large part of the genome. If you take this in aggregate, 1% of the genome are these little yellow parts, which are exons, 30% are introns, 70% is filler, that we really don't know what it does yet. That's what the human genome looks like. We care a lot about the protein coding sequence, all of these exons, because we know that mutations within these exons can absolutely lead to diseases like Duchenne. This DNA gets transcribed into a long RNA that includes the introns. Those introns get spliced out and removed so that you put exons together in order. So typically exon 1 goes to 2, 2 goes to 3, 3 goes to 4, et cetera. You make a final mature transcript. That mature transcript has some parts that tell the protein coding machinery to load on, and then to make a protein out of this sequence here, and then to stop, and then that's protein synthesis. Happens for every single gene in the genome. And so you've got then a different set of different mutation types that you can understand through that genetic code. So a mutation type that's about 10% of all DMD mutations are nonsense mutations. And in this example, a CAG, I go over to my code, CAG is including a glutamine. And if I change the C to a T, and I go back to my code, TAG, that means stop. So it means that the cellular machinery, as it's reading through CAG, 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 it's going to put glutamine, 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 glutamine in. But in this instance, when we change this one base to a T, it goes glutamine, glutamine, glutamine. I'm supposed to stop. And it doesn't matter what's back here because it stopped. So it's done, and your protein is dead. It will misfold, and it will die, and it is non-functional. That's called a loss of function mutation, and there's a variety of ways to get to that as well. Another point mutation type, and this is really, really unusual in Duchenne or Becker muscular dystrophy, is a missense mutation, but it is common in other genetic diseases, where in this instance, CAT, go over to my table, CAT encodes a histidine, so we get histidine, 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 et cetera. And in this example down here, we change the middle A to a C, so instead of it being a CAT, it's now a CCT. So instead of encoding a histidine, by changing that one letter in here, it goes to a CCT, which encodes a proline. So that's a missense, and it changes what the amino acid is, but the protein gets made all the way along its length. So it's generally a stable protein, generally a pretty good-looking protein. Small insertions, about 10% of our patient population have small insertions where you might just have one letter, an A or a T or a C or a G added in to the sequence, and that causes a frame shift. So in this example, we do histidines all the way through with our CAT, going along here, CATs. Here, we added in an A, but the cellular machinery is not very clever. It just has to read it in order. It doesn't get to know, oh, I should have just ignored that A and moved over. Instead, even though every base is correct from here on to the end of this gene, by adding this A in, I frame shifted the reading frame. So now it's ACA, the next one's a TCA, 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 and every amino acid is wrong till the end of the protein. In these instances, the reading frame always hits one of those nonsense mutations, and it just stops making a functional protein. So it's always a non-functional protein, and you can think frame shifting is always a broken protein. You can do that by inserting small bits. You can also do it by deleting small bits as well, just the opposite. You'll take a letter out, it'll also frame shift. This computer is quite lackadaisical in its willingness to move forward. Maybe I put some huge slide next. It's having to think a lot about it. Yep, there we go. So the other is you can also interfere with, oh, now it's a problem. If there's a little bit of sequence here at the end of every exon and at the beginning of every exon that says, hey, I'm an exon, join me together. So if you have a mutation in these bases right here at the edge or right here at the edge, it's obvious that you can't join 12 to 13. But there can be different consequences. Sometimes you can extend exon 12 forward. Sometimes exon 12 is missing. Sometimes exon 13 is missing. And it's a little harder to predict splice site mutations for exactly what their consequence is. But in general, they cause frame shifts. All right, I'm gonna have to talk a lot slower now in between each slide. There we go. So this is a reading frame map of the DMD transcript. So this is exon 1 here through exon 79 here. And I suspect many of you have seen this, probably even in clinics. This is actually fairly standard to describe it this way. So if one of the 3-letter codes ends exactly, so here within exon 3, say the CCT is here ending within exon 3, and then the next amino acid is encoded with exon 4, it's flat. If one of the bases of the next 3-letter code is starting in number 1 and ending in 2, it's written as an angle this way, and if the opposite is true here, where the first letter is encoded by, the first 2 letters are in 6 and the last letter's in 7, it's encoded the opposite way. So this is a pretty convenient way to look at reading frame. So you can start to say, boy, if I deleted exon 31, I wouldn't affect the reading frame. But if I deleted exon 8 or say 45, I would affect reading frame. How do I know that? Because this edge is not flat. So if I took exon 45 out and then focused on what was happening just in this area, if the simplest thing that I'd predict happened, if I'm genetically born without exon 45, which is the single most common mutation that we observe in Duchenne, exon 44 is joined to 46, and it would be out of frame. So everything past that point is going to be out of frame and result in a non-functional protein. So you can do this, and that's most of the people in the room and most of the people that you know with Duchenne have loss of function mutations because of a large deletion, typically an exon deletion, multiple exon deletions. And further, many of those mutations are between exon 44 and 45 is the core hotspot of where mutations occur. So this is sort of a density plot in this relatively small sample. Majority of the mutations, deletions, duplications were actually occurring in this region here. So reading frame is critically important in Duchenne. All of your clinicians are aware of this. All of your clinicians think about it. And so we had some instances when the gene was first identified of very large deletions leading to a disease where even in your 50s or 60s, you can remain ambulant and walking well, versus with much smaller deletions, you can end up needing a wheelchair by age 10, 11 with a more typical Duchenne muscular dystrophy disease course. So why can a big deletion cause less of a problem than a much smaller deletion? And that was largely explained by the reading frame rule, which means that if a mutation, if this matches to this and it remains in frame, you make a functional protein, and there's some function of that. If, however, this smaller deletion is resulting in an out-of-frame protein and a premature stop, you get no functional protein. So this is called the reading frame rule, and it's about 95% accurate, which is pretty good. But the amount of dystrophin that you make is actually critically important. So this is a boy with Duchenne, typically no dystrophin being expressed. There's a small island here of dystrophin-positive cells. And this is a man inadvertently diagnosed at age 27 with a Becker mutation, but he had no idea he had a muscular dystrophy. He was diagnosed because we inadvertently diagnosed his daughter as a carrier of this mutation because she was getting genetic testing for developmental delay. So a completely inadvertent diagnosis, and he had no symptoms whatsoever with this amount of protein being expressed. So the amount of protein you make is critically important. This is the dystrophin protein, and the way to think about it is as a shock absorber, is sort of the core way to consider it. So the beginning part has to bind to actin binding domains. The middle part is a bunch of hinges, like shock absorbers, and then the end part interacts with proteins at the cell surface membrane here. It's called the dystroglycan binding site. So if you're missing this, that's a problem. If you're missing this, that's a problem. But if you just shortened it up and made this the spring region shorter, but it was in frame, it's still a pretty functional protein. This is about 95% of the reasons for why some patients have a Becker muscular dystrophy-like disease presentation with large mutations in dystrophin, and others have a more Duchenne-like presentation. So There are exceptions to that reading frame rule, though. If you have out-of-frame mutations right at the beginning of the gene, you can sometimes have a relatively more mild disease course. So we're less certain of those bits of data. We're less confident that when we see an out-of-frame mutation that it really causes Duchenne muscular dystrophy. Also, there's some well-known mutations which are a little bit more mild, like an exon 45 deletion can be more mild. And sometimes in-frame mutations, when we're affecting this domain, actually lead to severe disease. So there are some reasons where we have to be cautious in terms of where we use DNA data to infer how severe the disease course is going to be, and we have to be cautious about those and sometimes wait to see how the child is doing over time. So this is an example of looking at that data. So this is a group of boys with exon 3 to 7 deletion, which is out of frame. It should cause Duchenne, but a number of those boys are remaining ambulant past their 15th, 16th, 17th, 18th birthday. So some of these young men do quite well with an exon 4 to 7 deletion. This is the typical disease progression in terms of age along the bottom and percentage who are remaining ambulant, hitting an average around 13 years currently with current care. But we do see some, like an exon 45 deletions, who are deviating from that, but not, certainly not typical Becker-like presentation, but they can have a more mild disease course. So why do some of these people have a more mild disease course other than the DMD mutation? There's a growing literature about other genes and their variations in those genes, like variations in LTBP4, which predict age at loss of ambulation in Duchenne. So we haven't learned about all these mutations yet in the genome, and that's one of the ongoing projects. So it's now possible. You can all go get your genome sequenced. You can actually just do it online if you'd like to. This is quite possible to do. So there's commercial vendors that will all sequence your entire genome. This has become quite routine. When I was first a postdoc, it was absolutely impossible. The human genome was proposed. We all spent $3 billion sequencing the first human genome. And now, in about a week's time, we can sequence any individual's genome for about $1,000. So it brings it to the very plausible way that we can do this on hundreds, thousands, actually ultimately millions of people. So there have been hundreds of thousands of people currently who've had their genome sequenced. Only a few hundred boys with Duchenne have had their genome sequenced, and we've been using that to try to find and look through all possible genes, how variations in them might be affecting the disease severity. So that's a DNA modifier project of Duchenne. So we've sequenced about 300 boys so far. We try to take boys that are at the relatively mild end of the disease spectrum, who are perhaps walking into their late teenage years, which happens with some of the true loss of function mutations. And then we also search for boys who are at the relatively severe end of the spectrum. And we do have boys who are using a wheelchair full-time at age 4, 5, and 6 years of age. So that does happen on the other end of the disease spectrum as well. So we search for those boys, and then we sequence these boys that are relatively more mild and relatively more severe. I don't have any data to share with you at this moment, but we're using whole genome sequencing to identify genes that are mutated that might make the disease more severe, or genes that are mutated that might make the disease more mild, and these can give us insights into how to modify the disease or how the human body has already modified the disease and what can we learn from that. So I was going to touch on a couple of other issues in terms of diagnostic issues. So I don't know if many people have had muscle biopsies done here in the room. It's actually relatively uncommon and not necessarily necessary much of the time. So when is a muscle biopsy needed in Duchenne? It is needed when no clear mutation can be identified by any of the clinical DNA genetic testing, typically done from blood. It's also reasonable to do if there's really striking and unusual deviation from the expected disease course. Either somebody is progressing much more fast than you'd have thought, or somebody who's actually having a relatively slow disease course, because that mitigates or might change how we would want to use things like steroids. In some instances, we also want to muscle biopsy females who may be having muscle weakness and we're going to presume that they might be manifesting carriers, but to prove that, we actually have to do a muscle biopsy. And then we do a fair number of muscle biopsies in the context of clinical trials. We've worked hard to try to mitigate and lessen the burden of doing muscle biopsies. So actually, my own son has never had a muscle biopsy, but he probably will soon. And there was never a good reason for doing it. But part of the reason I was resistant to it is this is what an open muscle biopsy looks like. This is one boy, had it on his biceps. This is a 2.5-inch long scar to get a tiny little piece of muscle out for a clinical trial. This boy at the end of this trial will have had 3 such muscle biopsies. This is what it looked like on the day of. This is a couple weeks out, and this is actually a couple years out. This is the scar that he's left with. It creates a substantial inhibition of people willing to perform muscle biopsies. In contrast, the new system that we've pioneered and then pushed forward is core needle biopsies. This is the scar actually 2 days after having done one on a boy who's now walking around just fine having done this procedure. And we inserted a needle here, a very big needle, I will admit that, but with local numbing into this young person's muscle and did the vastus lateralis biopsy here. And through that single hole, one could get as much tissue as we typically get from these and preserve it in a way that's actually superior to what we've been doing in clinical practice. So this is going to become the standard. It is not the standard, and many of you who are in clinical trials will need to be doing open muscle biopsies because of how the protocols have been written so far. But we hope to push the entire field to do this completely routinely, and I think it's going to change in the very near future. Oh, good Lord. It did all the additional. That's right. That's usually not my problem. Yeah, if you start anywhere in there, well, that's so slow. Just, yeah, start in that. Yeah, start in that one, please. So, oops. Aha. Thank you. All right, we're unprotected here. That's what that message reveals. So this is an old school muscle biopsy, and this is actually the way this needle was designed. There's a good reason for physicians never to adapt it to using this very routinely. This new system is actually designed for doing breast cancer biopsy samples, and it's just kind of a workhorse. And there's a little window here for taking a muscle sample in. The histology of the muscle is fantastic. And we can sample muscle relatively easily. So that's my diatribe on muscle biopsies. But what can we do with it as well? We now have some of the tools to actually diagnose and solve some of these boys that have actually been very difficult to resolve what their DNA mutation is. This is a boy. He came to us at 7 years of age. He had a characteristic Duchenne presentation, a very high CK. His del-dup analysis, sequence analysis of the DMD gene was normal. People kept sending it off because, what the heck, he's got Duchenne, got to go find the mutation. So that's what we do as physicians. We sometimes just reorder the same test. The person eventually got a muscle biopsy, an open muscle biopsy in this instance because it was a while ago, and it showed no dystrophin staining. So he really had a diagnosis of Duchenne, very clear, but there was no DNA diagnostic and no way to have a DNA test for anybody else in his family. So we took a little bit of his muscle, we sequenced his entire RNA of the muscle, and we looked at the DMD transcripts. This is exon 1 through 79, and what we saw was that after exon 37, there was no expression of exon 38 up to about 62. So even though there's some genetic material that's there from when the clinical testing is done, so it wasn't entirely clear what was up here, but when we went and shotgun sequenced his genome, so you can take little samples, this is the X chromosome here, we take little samples from all across the X chromosome, we could find some reads, which this is where the location of the DMD transcript should be, and this is about 5 megabases away, and these two things ended up being inverted. So just a big old change in the genome, 5 megabases just swapped around. No reason to have suspected it. It affected no single exon of the DMD transcript. They're all there. They're just in the wrong order. So what happens when we sequence the entire genome, we could see some reads here and some reads here, which span an abnormal breakpoint at one end and a different abnormal breakpoint at the other end, which was the result of an inversion. So his genome got put in backwards, so he starts making DMD this way, and then at this location, it stops making it because it's now 5 megabases away. So we can now find it, so we sort of solved it, but what's very gratifying is you can actually design a really cheap little simple test here and here to prove it in every other family member. So you can make now a custom DNA diagnostic that can be used in every family member and given a clinical report for this. So we can do this for every patient who fails to have a molecular diagnosis. We actually shockingly still don't solve everybody, but we solve about 90% of those that we have a muscle biopsy for. In this family, the small muscle biopsy lets us solve by RNA. Combined with the whole genome, we could solve this person, and then that gives us a diagnostic that we can use to go through his family, and then determine who is a carrier, who's not a carrier, and that ripples then through the entire family. Oh, actually, I'll make one other editorial. I don't know if this is relevant to anybody's family. Geneticists are often loath to test young potential carrier females, and I'm less loath to test young carrier females. And in this particular family, I actually highly recommended that we actually tested the young carrier females in this instance. And the reason for that is we just developed a completely custom DNA diagnostic today, a few years ago now, and this person, if they were told they might be a carrier and weren't connected to that exact DNA report, might go get genetic testing done that's out and available commercially. They will be told it's normal. It will be wrong. It'll be inaccurate. So these are instances where we shouldn't rely on kind of the core wisdom of medical geneticists in these special instances. So once we solved it, I felt compelled that we really needed to test everybody in the family so that it was in their medical record rather than they had to remember. For instance, this person here might have to remember, oh, my cousin had Duchenne, and maybe I'm a carrier because I don't know why. Maybe you could send me off for genetic testing. That will be wrong genetic testing. Editorial comment. Female carriers, I'll touch on a couple more issues before wrapping up. So female carriers of a DMD mutation generally have no clinical manifestations during their entire life that are of any substantial significance. About 70% have slightly increased blood CK, meaning some muscle is being damaged. Some of these women actually recall having mild muscle syndromes at a younger age, like cramping as a young child, and a small percentage actually have manifesting real muscle symptoms and having a progressive muscular dystrophy themselves. The important part here is we shouldn't necessarily always counsel people that all carrier girls will be completely unaffected. It's not true. And it's not the appropriate way to interpret all the data that we have. However, most females who are carrier mutations will not have any clinical manifestations. For female carriers, it's important to do a muscle biopsy, partly because if you had a high CK and you were a carrier of a DMD mutation, that doesn't prove that your high CK or your muscular dystrophy is because of that. So you really need to do a muscle biopsy in those cases, although in most instances, the solution will be that there's variable expression of the DMD protein. This does not want to move forward. So this is a woman who we biopsied just as we were testing out these needle muscle biopsies, and she was quite cooperative. But she has a high CK. She's a mildly manifesting carrier of Duchenne. She was unable to rise from the floor, now at 50. This brown stain around most of her muscle cells is dystrophin, and that's normal. Here's a muscle section, however, that has no dystrophin, and muscle sections like this, which have a nucleus in the middle rather than nucleuses at the edge, are all abnormal. So this is diagnostic of being a manifesting carrier female, consistent with her symptoms. So in carrier females, checking a CK is essential. X inactivation is commonly the cause of why this occurs, but sometimes the skewing that's in blood is not representative of what occurs in muscle. And again, we want to move to muscle biopsy. The final thing I'll touch on is newborn screening for Duchenne muscular dystrophy is going on as we speak. It's not universal. It will be universal. But at the moment, Perk and Elmer actually tested a bunch of kids here in California developing a blood spot test. So they took boys that were born within 10 years of when they were doing this study, consented them in a variety of our clinics, collected their blood spots, which are available on all of our kids in California. So for many research projects, we can go and do this sort of thing. And then they were able to develop a very highly specific test just on CK on that blood spots. These are the diseased kids, and this is all bunches more normal kids. There's a good separation of that. So the idea is you can take this window at some specific cutoff, and above that, you'd go on for further genetic screening. So this is actually in a pilot phase right now. So it's a pilot project to test 100,000 newborns over the next couple of years in a part of New York. And the expected hit rate will be about 1 in 10,000 live births. So summary from all that, thank you for your for receiving your genetics lesson today. I hope that all of you understand that your DMD mutation is actually incredibly important to know. It will become more important in the future. Most of your mutations in DMD, it doesn't matter what the mutation type was in terms of the phenotype. That is, it's a loss of function mutation. If no mutation is found, I wouldn't let the ball drop. I'd move to this next step. It does need child needs a muscle biopsy, and there's some advanced diagnostics that can actually lead to creating a custom DNA diagnostic for a specific family. Core needle biopsies are going to become a more routine part of what we do for diagnostics moving forward, and I hope to make it and have it be a more common and routine part of what we do in clinical trials. I hope the world across many different centers will be able to show more genes that are being modified, that are modifying DMD severity, and we will be living through the era of newborn screening as we speak here in the next few years. These are acknowledgement slides and some of the sources of funding that we rely on at UCLA. Thank you so much.
You want to grab Katie? We'll open it up for questions here. I've seen quite a few hands pop up already.
Sorry, I had my back to you the whole time.
No worries. So thank you so much. Question for you, and that is, I'm a genetic counselor, so just a very quick question about the very last thing you said, the newborn screening program. Is there enough of a distribution separation for your patients with high CK that you can figure out it's Duchenne versus Morosin deficient muscular dystrophy or limb-girdle muscular or any of the other ones? Or is it, no, if it's high, now we start down the pathway of what is this?
Yeah, so the way this program will work is it'll be a high CK. It'll be around 700 to 1,000, and that will screen out almost all normals. But even in the plot that I just showed you, some normals will still be caught. So people will certainly be repeated for a CK level if persistently high. The number of diseases that cause a CK of 20,000, 30,000 is really small. So I think true Duchenne in the newborn period will be picked up very well by this. Certainly you'll be sequencing a larger group of people who will be negative for DMD mutations. So that will happen. We'll capture people, actually we'll even capture some normals potentially, or people just with hypercemia, which happens and we don't know about them for their lives unless some doctor bothers to check a blood test, right? So there's a variety of reasons. It'll be just like all other newborn screening, not specific for the big screen, but specific for the final. So I think we'll have very few false positives for once the DMD gene sequencing is done. But I think it'll be important, again, like other newborn screening, not to panic parents just because of a high CK at birth. That is not diagnostic of Duchenne.
Has there been any association with gestational age and CK level, or I'm assuming that's probably factored in?
No.
So for kids who are born premature, for example.
Yeah, no, but in general, they all have CKs in this kind of range. You saw the smear there was quite broad. So there's a higher CK kind of in that newborn period, but the CK that we see in Duchenne or even Becker is, so far off, it's, it's 20, 30 standard deviations away from normal. So it's wildly different, yeah.
Oh, I guess I had a question. You were talking about checking CK levels for carriers? Because I was recently just had a blood test and found out I'm a carrier. Are you saying that I should have my CK levels should be tested?
Yeah, yeah. So most women will have no symptoms of muscle disease at all, but one of the signs of the muscle disease will be a high CK. The reason that I test all carrier women with a CK is it can also have you can sometimes have a fairly high CK without any symptoms whatsoever. So you can be leaking this CK, which is creatine kinase, which is a protein, an enzyme inside your muscle. When that happens in our boys, actually, I'll bet somebody in this room got a liver biopsy for their diagnosis of Duchenne, right? That happens. It happens because AST and ALT, which we teach in medical school, or we even call them liver function tests, they leak out of the liver. So the same thing can happen for carrier women. So we've actually had instances where our Duchenne boy was diagnosed because his sister's a mildly manifesting carrier with high CK. She was tested. She has high liver function tests because those enzymes are also in muscle. She got a liver biopsy. They kept working her up for a liver disease until it was figured out that really her brother has Duchenne. She's a mildly manifesting carrier. So part of the reason to do it is to know what it is, know if it's an issue, and make it so that you don't inadvertently go down a wrong track across a problem that we see persistently in neuromuscular diseases.
So I could just request that through my regular
Yeah.
primary care physician?
Yeah,
Okay.
yeah. And often it's more likely to be high if people complain of weakness, particularly in the hip girdle area, or have cramping. So.
Hi, just have a couple questions. I know medicine has gotten pretty good these days. And you said there's a newborn testing. Is there a testing while the baby's still in the womb?
Yeah. So only if we know the mutation, though. So if we know the DNA mutation, we can do, so many of our families at UCLA and other medical centers, when we figure out the mutation, we can actually make a DNA probe, even if it's for this family here that I just described, that can be used for pre-implantation genetic testing. So you can do in vitro fertilization and actually choose embryos that are devoid of a mutation if that's what you chose to do. It's possible to do during pregnancy by either chorionic villus sampling or amniocentesis by simple DNA testing. So most of that DNA testing is a simple PCR-based analysis once you know what the mutation is. So.
Okay. And is it, when you find a boy with Duchenne's and then they get their siblings tested, is it assumed, or assume's a terrible word, but would they have the same? Would the siblings possibly have the same mutation as the boy, or would there be something different?
So almost always. So again, everything weird in the world has probably happened somewhere. So almost always, a mutation that occurs in a family is the same mutation, and everybody that's directly related in that family always has the same mutation. However, there's one family that I was made aware of who I was guilty of resending their genetic testing because brothers had two different mutations. So both affected by Duchenne, slightly different mutations. So, the lesson and the way I would think about it is there's nothing about a DMD mutation that protects you from everything else that can go wrong, right? And that's the same as actually another DMD mutation, right? So all those things are possible to occur, but they're super rare. And that one lands in this realm of, 1 in 5,000 risk of a DNA mutation at any location. The next egg over also has a 1 in 5,000 risk. So if we look at 25 million boys, one's going to have, some set of families are going to be like that. So it does happen.
Okay, thank you.
I'm sorry, the other corollary of that is because that does happen, it's reasonable to test every family member. Okay.
Here's another family member.
Then you need to be tested.
Yeah, well, yeah.
No, yeah.
Yeah, but his mother's not my mother, so.
Fair enough.
So would doing a genome sequencing of Michael help?
So I would not say it would help, but I do think there's, when we have kids that are on the relatively more severe end or the relatively more mild end, they contribute more information to a genetic study. So
Okay.
the idea is we're trying to sequence whole genome, sequence every single gene, categorize every single mutation that occurs. The one way it's a practical help is we do look at the DMD gene, so we know exactly what the mutation looks like, but rarely does that help because usually the clinical diagnostics are completely accurate and there's nothing new that we add to that when we do whole genome sequencing. So I wouldn't encourage people to necessarily go get whole genome sequencing, except for in this kind of context of doing this, advancing the research for why we wanted to figure it out, yeah. There will be a world where I believe we will want to know not only the DMD mutation, but like much of the rest of medicine, we'll want to know what mitigates that risk. So if it's encoded in your genome, we're going to be able to decode it and report it. So if we knew all sorts of genetic modifiers that predicted somebody was going to walk until 20, as a physician, I'd like to know it. As a parent, I'd like to know it. And I'd like you potentially to adjust how you think about steroids in me. Conversely, if I have something that's going to be much more aggressively progressing, I might be much more willing to try other therapies or be more aggressive with therapies. So practical ramifications.
And then, and I'm going to ask a question. It's probably not couched right, but I was trying to write it very quickly as you flip to another.
As I tried to flip to another slide.
Well, this was early on, so you were doing well at that point. Can blood molecular analysis determine if a treatment, such as Adalurin, will work better or anything like that?
Not better, but some of the treatments are not relevant for some mutations. So take the Adalurin example. There'd be no reason to try Adalurin if it's a frameshift mutation that hits a nonsense mutation in a few of these 3-letter codes. That nonsense mutation will not be helped by Adalurin because it'll still be out of frame. It's still a nonsense protein. But if it's just the first example that I gave you, if it's just a nonsense mutation, just changing a CAG to a TAG, those are the types of mutations that are the ones that are eligible for a bit of a gene repair by Adalurin. It wouldn't predict whether it'll be a better amount of dystrophin made or a lesser amount of dystrophin. It won't predict for us yet why some people might respond better than others.
So we should talk to our geneticist to find out what type of code.
Yeah,
And-
I would encourage all of you to understand it completely.
Completely.
It's not much more complex than what we described. I think there's excellent resources. All genetics counselors know these things backwards and forwards, and I think there's a really good reason to know it in Duchenne. And sort of many of us on the clinical side, it's what's interesting is in Duchenne clinic, sort of the discussion of the mutation is very robust because it matters, right? So we even think of the patients as distinct mutations as a part of what their phenotype is. And that's actually relatively rare in the rest of our genetics practice. It's, you got a mutated gene, they've got this disease, versus here it's many, many different, thousands of different types of mutations all lead to the same disease, which is Duchenne, from the same mechanism, but it leads to different therapeutics that we're already seeing.
Any other? John.
Thank you. Just to clarify my brain a little. So no one else has been tested in my family. I've got another son. He's 25 years old and strong as an ox.
He doesn't need to be tested.
He doesn't
then.
have to worry.
Nope.
But
No.
my wife should probably, starting with the CK,
CK is easy. I would get the CK and then the blood test for the DNA. So the CK is something that the field used to use before we figured out the DNA So you'd use CK as a proxy of the mutation, and sometimes you'd even use a closely linked marker to figure out did the mutant chromosome come down, but only when you had it running in your family were any of those things useful. She should get a CK, and she should have the mutation that's observed in your son tested in her. Yep.
Okay.
Yeah.
And the problem could be she could be improperly diagnosed with a liver disorder?
There's a risk that if she came in, she could come into a medical practice and somebody will test her for her blood. And one of the common things that we do when we send off a comprehensive metabolic panel is it'll look at kidney function, it'll look at metabolic function, it'll look at some of the breathing studies that the respiratory doctor was talking about. And it will often look at liver function because there's a lot of liver disease out there. There's a lot of reasons you can catch hepatitis, right? Infectious hepatitis and chemical hepatitis. So it's a very common thing to look at. And it's so much more common than Duchenne or being a Duchenne carrier female that it won't even be on the radar even when she goes, right? So it's just something, it's a small prophylactic thing to do.
Thank
Right.
you.
Sure.
Oh, I guess I just want to make a comment. Yeah, for carrier testing now, you can go through Parent Project Muscular Dystrophy
Yeah.
is it, well, whatever, will pay for the testing, where sometimes I had trouble before where insurance wouldn't cover the testing. They will cover the testing if you have a son that's confirmed, they will cover
Yeah,
the testing
thank
for
you for pointing that out. Yeah. So, and it is actually, that's a slightly different editorial comment. It's absolutely insane that our insurance companies are not forced to cover carrier testing for a disease like Duchenne, but it is common practice. And actually some insurances are not even mandated to cover genetic testing, so, yep. And even our own Medi-Cal, when we have people on Medi-Cal in the state of California, as wealthy as we are, a child who's diagnosed with Duchenne does not mandate testing their mom, that we can often have untested mothers from that. So there's a lot of gaps still, and these testing programs are critically important. Thanks.